Assalamualaikum warahmatullahi wabarakatuh.
Walaikum salam, sir.
We are working on the metabolism lesson. Today we have to discuss the portion called the regulation mechanism. Regulation mechanism of enzymes. It is the portion we have to discuss. Very cold. Okay. Right. So in the regulation mechanism, right, the activity of a, right, here we first introduce a kind of enzymes. Remember these kind of enzymes. Now let me introduce it. Right. Regulation mechanism of enzymes. In the regulation mechanism, they are explaining you how many headings, 3 headings. Allosteric regulation of enzymes, allosteric activation and inhibition, and feedback mechanism. First of all, I need to introduce you, right? Allosteric enzyme. What is an allosteric enzyme? If we have a clear idea regarding allosteric enzyme only, we can further proceed in this particular portion. Remember, all the enzymes are not called allosteric enzymes. Okay, one set of enzymes are there who can be called allosteric enzymes. Right. I will give you a definition to an allosteric enzyme. Right. An allosteric enzyme is an enzyme that contains a region to which a small regulatory molecule binds in enough. Fine. Please take it down.
Done, sir.
An allosteric enzyme is an enzyme that contains a region to which a small regulatory molecule binds. Okay, I am going to introduce you an allosteric enzyme's nature. Normally, okay, allosteric enzymes contain more than one, okay, protein subunits. Mark this point also. Allosteric enzymes contain more than more than one protein subunits. Okay, what is an allosteric enzyme? Is an enzyme that contains a region to which a small regulatory molecule binds. Second, allosteric enzyme contain more than one protein subunits. Normally, there will be more than one protein subunit. Okay,
Okay, sir.
right. Mark this one also. In each subunit, there will be an active site. In each subunit, there will be, what's it? An active site. Every subunit contains an active site.
Done, sir.
Right. Another point regarding it. Another point regarding it. The allosteric site, that is the site to which the regulatory molecule bind is situated between the, what is it? Between
Subunit.
the subunits. Mark that point also.
Done, sir.
Right? Have you marked it?
Yes, sir.
Everyone, have you marked it?
Yes, sir.
Good. Now, with all these features, let us introduce the allosteric enzyme. Okay. I am just drawing a simple allosteric structure. In this allosteric structure, say this is a allosteric subunit. Okay. This is another allosteric subunit. No need to be in different colors, but it's easy for me to sketch the borders. Right. So here I have taken 4 subunits. One subunit, another subunit, another subunit, another subunit, all together form a, what is it? Allostatic
Enzyme.
enzyme. In the allostatic enzyme, okay, as I have pointed you, there will be each having an
Active site.
active site. Here the, what is it? Active site.
Active site.
Understood?
Yes, sir.
Right. This is the, sorry. This is going to be, these places are there, no? Can you notice?
Yes, sir.
This is the, these are the places what we call by the name allosteric sites. Okay, these are the, what is it? Allosteric
Allosteric.
sites, right? To that only the regulatory molecule will bind. Clear?
Yes, sir.
Right. Could you, so what are the components? What is this?
Protein subunit.
Protein subunit. What is this?
Active site.
Active site. Those things are there. Between the subunits, there are places for the binding of the regulatory molecule. Okay, allosteric site is joined by a regulatory molecule. Could you please draw this kind of a diagram and label the components?
Done, sir.
Completed? Have you all completed it?
Yes, sir.
Good. So, here I am introducing you a similar allosteric enzyme. 4 subunits, correct?
Yes, sir.
Each having an active site. in between the active site, there are regulatory site.
Molecule.
Can you see? Okay. Remember, an allosteric enzyme doesn't need to have all the time 4 subunits. Here, the example we are taking has 4 subunits. More than 1 subunit. That is the idea. Okay. Right. Now, another property of allosteric enzyme we are going to speak here. That is which property? Which property?
Oscillation.
Oscillation property. Oscillation property, what the enzyme does is, enzyme, okay, oscillation mean moving, correct? Moving to this side, then now in our mind, let's say you are going to do a work, your mind says do, again your mind says don't do, again your mind says do, again your mind says don't do, don't do, don't do, don't do mean, we used to say the mind is oscillating. Understood?
Yes, sir.
The mind is oscillating. So this enzyme is oscillating between how many forms?
Inactive form.
2 forms. How many forms? 2 forms are there. What are the 2 forms?
Active and inactive form.
Active. So will it permanently stay in active form?
No.
No. It now like the mind do, then the mind says, what is it?
Don't do.
Don't do. The mind doesn't stay continuously in the don't do. Again, mind says
Do.
do. Again, mind says don't do. Like that, will this enzyme permanently stay in active form? Mean no.
No.
From active form, it will shift to
Inactive form.
inactive form. From inactive form, it will again shift to
Active form.
active form, active form, inactive form, active form, inactive form. That way, there is an oscillation. Understood?
Yes, sir.
Right. If we want to get use of this enzyme, it should be continuously in the active form. Yes or no?
Yes, sir.
If you want to get use of this enzyme, it should be continuously in the, what's it? Active form. So what's happening is, when it comes to the active form, we are not bringing it to the, what's it? Active form. It will normally come to the active form. Yes or no?
Yes, sir.
Only problem is it will not
Stay.
stay.
Right.
It will come to the active form, but it will not stay in the active form. The only thing we have to do is, when it comes to the active form, we have to stabilize it in the active form without going to the
Inactive form.
inactive form. So, when it comes to the active form, in the regulatory site, an activator molecule binds. When it comes to the active form, in the regulatory site, Who binds? Who binds?
Activator.
Activator binds. When the activator binds, one regulatory site is enough. When the activator binds, then it will stop its oscillation nature. Understood?
Yes, sir.
Are you clear? When
Yes,
the
Yes, sir.
activator binds to the regulatory site, the enzyme will stop its
Oscillation.
oscillation and will stay in the
Active form.
active form. Right? Then we can get benefit of its catalytic activity. Clear?
Yes, sir.
If you look at this diagram, you can see the activator molecule has joined between the two subunits, what is it? A and B.
B.
Am I right?
Yes, sir.
C and D doesn't have anything direct with the activator molecule, but they have interaction with A and B, correct?
Yes, sir.
Yes, sir.
Activator molecule contact the A. From there, the signal goes to
DNC.
DNC. So they also stop changing their shape. Entirely, all the subunits are stabilized with one activator molecule. Clear?
Yes, sir.
Right. There are instances. Now say you are taking a medicine. Say you are taking a medicine. The medicine is trying to stop the function of an enzyme in the body of a bacteria. If that enzyme functions, bacteria will perform. If bacteria performs, we will get the disease. Understood?
Yes, sir.
So here, an inhibitor, medicine does the inhibitor work. Does the inhibitor bring the allosteric enzyme to inactive form? Tell me. Now this story is over. Active form story is over. I am withdrawing from the, what is it? Active form. I am again taking, there is an allosteric enzyme. It is oscillating between whom and whom?
Active and inactive.
Active and inactive form. If it is stabilized in the active form, it will be catalytic. If it is stabilized in the inactive form, it will be non-catalytic. Understood?
Yes, sir.
Yes,
There
sir.
are instances the enzyme should be turned to, what is it? Non-catalytic. Sometimes permanently, sometimes, what is it?
Temporarily.
Temporary.
Temporary. Understood?
Yes, sir.
Sometimes permanently, sometimes temporarily. Now, in certain things, when we use some device, okay, we will keep some device without using. Okay, let's stop using this one for a while. We have that one, no? Correct? Some device, some equipment we are using, we stop using. No, don't use it for this less much time. Oh, use this one for this much time. So see, that is their similar work. So when the allosteric enzyme comes to the active form, if you want to stabilize it in the active form, who should be joined to it?
Activator.
Activator. Correct? When the allosteric enzyme comes to the inactive form, to stabilize it in the inactive form, who should be connected to it?
Inhibitor.
An inhibitor. Are you clear regarding allosteric activation and allosteric inhibition?
Yes, sir.
Good. Certain. Okay. So let's take the allosteric enzyme. Right. 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 Here, if you take this note, the first point says, some points they say now, the regulatory site, a binding site in the enzyme other than active site is, what is it? Allosteric site. Allosteric site is a regulatory site. Do you agree with it?
Yes, sir.
Yes. A molecule joining the allosteric site can change the shape of the active site. That can happen. That can happen. But that is not fitting to the allosteric enzyme. Understood? Understood?
Yes, sir.
Are you confusing? Are you confusing? The second point.
Can I explain it that again?
Okay, sure. Now, this is the thing. There are two things there. Okay. This is speaking not about the allosteric. Now, what I explained you, allosteric activation, allosteric inhibition, that is not explained here. Understood? Understood?
Yes. Yes,
Yeah.
sir.
It is
Yes.
explaining. Now, I told you when the enzyme come to the active stage, an activator molecule bind. Does the activator molecule bring it to the active state?
No.
No. Adu oru mass. That's why it is called activator molecule. Understood?
Yes, sir.
Yeah. Other side, another thing can happen to the allosteric site. Okay. This is allosteric site. This is, what is it? I am taking a enzyme. Okay. I am taking an enzyme. This enzyme has, I have only 2 to 3 diagrams to draw. I'll manage. This enzyme has this kind of an, what is it? Active
Active.
site. Okay. There is a molecule. There is substrate molecule. Its substrate molecule. Just an idea, where its substrate molecule is like this. Okay. Its substrate molecule is like this. Now, are they fitting?
No.
No.
No, not fitting. So, a regulatory molecule or an allosteric molecule binds to, okay, an allosteric site here. Who is binding?
Regulatory molecule.
A regulatory molecule is binding here. Because of this binding, this shape is getting altered, okay, to this shape, okay, fitting with the home
Substrate.
substrate molecule. Here, does the regulatory molecule adjust the shape? Tell me.
Yeah.
Yeah. That is allosteric regulation. Allosteric. The allosteric. So who is the allosteric molecule here? Who is the allosteric molecule here?
The blue color.
The blue color. Which is the allosteric site here?
The place where the blue.
The place
binds.
where it is binding. Okay. If you remove this component out, this shape will be lost. Understood?
Yes, sir.
So tell me here, is this molecule responsible for this shape?
Yes.
Yes. Agree with it?
Yes, sir.
Yes. Same time. Okay. To the same enzyme molecule. Okay. To the regulatory site. Okay. If an inhibitor binds, who is binding?
Inhibit.
Inhibitor binds, then the shape of the active site will be changed, unsuitable for the home
Substrate.
substrate. Understood?
Yes, sir.
Right. So, this is not the allosteric regulation, the activation and inhibition I told you, right?
Yes, sir.
Here, the regulatory molecule decide in what shape it should be. Am I right?
Yes, sir.
Okay. Here, it can bind, correct? Here, it can't bind. Understood?
Yes, sir.
So, in this moment, what kind of molecule is this? Inhibitor molecule. This is active site. Clear? Sorry, not active site. Allosteric site. So this is the suitable geometrical shape of the active site. This is the unsuitable geometrical shape of the active site. Am I right?
Yes, sir.
Right. So could you please take this one? Okay, what is the heading? Allosteric, what is it? Regulation.
Done, sir.
Okay, right. So here active site shape is decided according to the regulatory molecule. Understood?
Yes, sir.
But the previous thing what I told it is, that is true. That is there. What I told you is there. This is one kind. Okay. One molecule join. It can be a cofactor. That molecule joining, the blue molecule can be a, what's it?
Cofactor.
Cofactor. Because of a cofactor joins, the active site is coming to the correct shape, fitting with the substrate molecule. Because a non-competitive inhibitor binds, the active site is coming to a different shape, not fitting with the
Substrate.
substrate molecule. Clear?
Yes, sir.
Right? So here, the regulatory molecule decides the
Shape of the active
shape
site.
of the active site. Yes or no?
Yes, sir.
Yes. If so, please go through the page number 14, allosteric regulation. Read and see whether the idea I conveyed you and the idea written there matching.
Okay, sir.
Are you clear?
Yes, sir.
Good. Everyone, is it clear?
Yes, sir.
Yes.
Yes. Second, it is the one I told you regarding the subunits, the oscillation, active form, inactive form, activator binding, inhibitor binding, stabilizing in the active form, stabilizing in the inactive form. All these things I told you. That is regarding the second component. Okay. In this kind of a regulation, does the allosteric molecule determine the shape of the active site? Tell me,
Yes.
No.
in this one, does the activator molecule determine the shape of the active site?
No.
No. Enzyme come to that stage, we catch it there. Understood?
Yes, sir.
When enzyme comes to the active form, we hold it in the
Allostrophic site.
no, we hold it in which form?
Active.
Active form. Okay, without moving to
Inactive.
inactive form. My question it is, in this one, the active site shape, is it decided by the activator or inhibitor?
No.
No. But
No.
in the previous one, if you take this one, active site shape is determined by the
Inhibitor. For the active.
inhibitor or regulator. Correct?
Yes, sir.
Can you understand the difference?
Yes, sir.
Okay. Now almost there are, it's not a good example. It's not a good example. There are, but it also can be an example. Okay. Think like this. Now you children are studying. You are doing your advanced level. Okay. Now I'm working in several different projects and meet different students. Okay. Some students are there. They have registered in a school and studying in another place. They have registered in, what is it?
In
One
a school.
school. Okay. In that school, they don't go for any educational one. No teachers are teaching them. Finally, they study somewhere. Finally, they write their examination, okay, through that school, and the results comes to that school. Understood?
Yes, sir.
So the school celebrates the performance of the child, but the school doesn't have anything, correct? Understood?
Yes, sir.
Sir.
When the child achieves it, because it has given the opportunity to sit the exam through that school, the school celebrates the victory, correct?
Yes, sir.
Certain schools are there, okay, they fully hard work on the children, day and night they work on the children, and they bring the result and they celebrate. The second case. In these two, I told you 100% that example is not matching, but just a match. Okay. In these two, which one is like the first case?
The second example?
The second example, correct?
Yes, sir.
The second example. Enzyme itself come to that stage, we catch it. That's all we do, correct?
Yes, sir.
Okay. That is like the second example. The school hard works, the school put every effort on it, change the child's performance, adjust the child, and finally the child brings results mean that is like this page number 14. Correct?
Yes, sir.
So it's not a good example, just an example. So can you understand the difference between this heading, allosteric regulation, and allosteric activation and inhibition?
Yes, sir.
Everyone, can you understand it?
Yes, sir.
Please go through the page number 14 under activation and inhibition. Only one thing, there is an example given to you. That example I will explain you. Okay. Rest you go and see whether things are okay, can understand.
Sir, can you explain the last point?
Last point mean the ADP, ATP, no?
I know, sir, the
Uh.
subunit point.
Amma?
The subunit point, sir.
Yeah, that's what I told you. This one I told you. What's it? Here, the activator molecule directly contacts between with whom and whom?
A and B.
A and B. Okay. A and B, when they receive the activatory signal, that signal is transferred to whom?
C and D.
C and D. So their active sites also come to the stable form. Understood?
Yes, sir.
They are not directly communicating with the activator. They are communicating with the
A
other
and B.
subunits, Correct?
Yes, sir.
The message they receive, you stabilize. A is stabilized, then A, now then that is general, no? That is very general, right? When you, now for example, you go for a class, okay? When you go for a class, when you see the teacher, the sir is good with you, then you will be activating the friends of you, correct?
Yes, sir.
I am going to this sir, you also follow, that is there. So that child doesn't have an experience with that sir. But that child gets the signal through you. Anyway, that child is also activated. Am I right?
Yes, sir.
So that way. So they have signal transmission among them. One subunit, if it is activated, all the subunits will be, what's it?
Activated.
Activated. Clear?
Clear, sir.
And the speciality it is, it's quickness. It's, what's it? The
Quickness.
speciality is its quickness. Okay, when one subunit gets the signal, quickly all the subunits will get the, what's it?
Signal.
Signal, and all will come to the
Stable.
position, in the activated position correctly. It's, I used to tell, don't mistaken me, keeping you telling like this. I used to tell, here subunits are like women. Understood? Subunits are like women. In 2018, I met with an accident in my car, a mass accident. With Allah's grace, I have not got any injury, but my car totally lost. It was my driver who was driving my, what's it, car, and he got arrested in the police. I went to Putlam. On my way only it happened. Okay. And anyway, I reached Putlam. I was staying in Putlam. Next day, I have to go for the legal procedures. I have to take my driver out of the police. I have to claim the insurance of my car. So there was a procedure going on. Meanwhile, I was listening, a spreading voice message from a teacher, from a, what is it, teacher speaking. She is speaking. Mr. Rifan, the science teacher, he was speaking. He got arrested by the police, keeping the driver on one side seat. He has driven the vehicle. Mr. Rifan has got arrested. Like this, he has created a story and she was spreading the story. Understood?
Yes, sir.
I was laughing staying in Putlam. My mama was calling, Rifan, inga odi vandu paru. Unna patti enna varudhu appdinu solli. You know, during, nowadays we have this WhatsApp, Facebook, like so many social medias, no, to spread a message. During the time of Hitler, okay, to spread a message all over the country, okay, he had a women team. He had a, what's it, a women team. That much quick spreading. Gossip. Nowadays males are also little by little, what's it, entering that field. But he can't beat the women. If you tell good or bad, quickly spreading. Understood? So telling to women this one sometimes might be, oh my God, sir is telling that we are spreading gossips mean I'm not personally attacking you. Understood?
Yes, sir.
My mother is also a woman. My wife is also a woman. Understood? I also have two daughters, girls. Truth is always bitter. What shall we do? So, like that, if one subunit gets the signal, it will be alarming all the subunits quickly, then all the subunits will be coming to
Active form.
active form. Clear?
Yes, sir.
Yes, sir.
That is one kind of nature. You can understand this allosteric activation inhibition nature and this nature are not same. Yes or no?
Yes, sir.
Right. That is one story. This is another story. Another story is that is before that one we will speak about this particular process. What is it? In catabolic metabolism, ADP function as an activator and ADP function as an, what is it?
Inhibitor regulating substance.
Inhibitor regulating substance. Here, right? When now say during, we speak about like this. I'm gonna எழுதுறது கஷ்டம். அங்க போய் எழுதுறேன். Okay. Right. Now, say in a cell, okay, you know, ATP's synthesis, correct? ATP, ADP joins, what is it?
Inorganic phosphate.
Inorganic phosphate and form ATP and what is it? Water. In this reaction, okay, there is an enzyme working. I already gave that enzyme. What is that enzyme?
ATP.
ATP synthase enzyme. It's an allosteric. It's an allosteric enzyme. Okay, to this allosteric enzyme, ADP works as an home.
ADP activator.
No, ADP works as an
Activator.
activator. ATP works
Inhibitor.
as an home. Inhibitor. Activator's role play it is, okay, due to the activation, ATP synthesis, what happens? Increases. Correct? If due to inhibition, what will happen? ATP synthesis, what is it?
Decreases.
Decreases. Why it should decrease? It should decrease only due to one reason. Okay? This is mainly when surplus ATP is, what is it? Present. Surplus ATP is present. Normally that's happening, right? Say there is a biscuit company. If they have sufficient stock, will they further and further produce the biscuits? Huh?
No.
No. They will wait, correct? until its use up. So, for ATP synthase enzyme, ADP works as an
Activator.
activator and ATP works as an inhibitor. Clear?
Clear, sir.
Yes.
Right. Fine. So that point is given to you. That point says you who is working as an activator. Who is working as an activator?
ADP.
ADP working as an activator. ATP working as the, what is it?
Inhibitor.
Inhibitor. Are you clear?
Yes,
Clear,
sir.
sir.
Good. That is one story. With that story, allosteric portion is over. Allosteric activation and what is it? Inhibition is over. This is another regulatory mechanism. Here it is very, very special mechanism. Very, very special mechanism. Okay. The special mechanism here is, here there is no any external regulatory molecule. Understood? There is no any external regulatory molecule. Substrate is the regulatory molecule. In cooperativity, who is the regulatory molecule?
Substrate.
Substrate is the regulatory molecule. You children know. Substrate joins which place?
Active site.
Active site. Is there any other site needed for the substrate to join?
No.
No. Okay. Look at this picture. Simple idea. Okay. Here also the enzyme is oscillating between which two form?
Active and inactive.
Active form and inactive form. When it comes to active form, substrate go and bind to it. One substrate, say, when one subunit come to the stabilized form, then automatically it will give the signals to
Stay stabilized.
the other subunits. All will becoming
Stable.
stabilized. Understood? So there's an important difference there. Here, the regulatory molecule is whom?
The substrate.
The substrate molecule. That substrate molecule is binding to
Allosteric site.
allosteric. That substrate molecule is binding to
Active site.
active site. No need of
Allosteric site.
allosteric site. Understood?
Yes, sir.
Binding of the substrate to the active site activates all the subunits to be in the correct form to accept other substrate molecules. Clear?
Yes, sir.
Are you clear? Can you understand the difference between allosteric regulation, inhibition, and this cooperativity? In
Yes, sir.
cooperativity, who is the regulatory molecule?
Substrate.
Substrate. Substrate binds to whom?
Active site.
Active site. If one substrate binds to the active site of one subunit, what happened to the other subunits?
all get stabilized.
All get stabilized in their active form. Correct?
Yes, sir.
Right. Here, this kind of a nature. Now, how look at this picture. How many subunits are there?
4.
4. Correct? When your examiner collected an example, your examiner collected the example of whom?
Hemoglobin.
Hemoglobin. Hemoglobin well fits with this idea, but remember, hemoglobin is not an enzyme. Understood?
Yes, sir.
Hemoglobin is not an
Enzyme.
enzyme, but rest of the things are well fitting with it. How many subunits are there in the hemoglobin?
4.
4 subunit. Who is the substrate binding to the hemoglobin? Oxygen molecule. If one oxygen molecule bind to one subunit of hemoglobin, all the other hemoglobin molecules are stabilized to accept oxygen molecule. That idea is there. But hemoglobin, is it an enzyme?
No.
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 So, what your examiner tells is, finally, cooperativity similarly works in, what is it? Multi-subunit
Enzymes.
enzymes too. Like that, he is closing it. What we point here is cooperativity is applicable to, okay, molecules other than enzymes too. Understood?
Yes, sir.
Enzymes showing cooperativity, true. Okay, hemoglobin-like components who are not enzymes also show, what is it?
Cooperativity.
Cooperativity. Examiner will catch that point. Examiner will catch that point and put you the questions. Understood?
Yes, sir.
Right. Mark this point and keep. The other thing. Wrote it. Adhan is over.
Done, sir.
Yes, sir.
Right. That is one kind. Cooperativity. Speciality here is substrate molecule working as the regulatory molecule. It is actually an activator. Substrate molecule never function as the inhibitor. Understood?
Yes, sir.
Right. Last, it is simple one. What is it? Feedback inhibition. In the feedback inhibition, now see, this is not the syllabus example, just a diagram, okay, to give you an idea, right? Now, can you see this is a metabolical pathway? What is the initial substrate? Tell me. 309. Not in syllabus. Don't worry, not in syllabus. Understood?
Yes, sir.
309. Here the product is, what is it? Isoleucine. Okay. The product is
Isoleucine.
isoleucine. Substrate is, what is it?
309.
309. Now, the total purpose of this metabolical pathway is converting the 309 to, what is it? Isoleucine. Here, enzyme 1 converts 309 to an intermediate called A. That intermediate A is converted to intermediate B by another enzyme. That intermediate B is converted to intermediate C by another enzyme. That intermediate C is converted to intermediate D by another enzyme. And that final product is home. Final product is home.
Isoleucine.
Isoleucine. As I told you, you don't need 309 or isoleucine in your syllabus. Now this end product, who is the end product of this pathway?
Isoleucine.
Isoleucine. It is binding with the enzyme 1 as an inhibitor. In what form it is binding?
Inhibitor.
As an inhibitor. Because it's binding, active site is losing its shape. Correct?
Yes, sir.
Right. When the active site loses its shape, can the enzyme 1 function?
No.
No.
If it is not functioning, can another metabolical pathway be performed?
No.
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 Until isoleucine is used up, it holds like this. After it is used up, enzyme comes to normal. It is relieved from the inhibition. Now it will be catalyzing another pathway. Understood?
Yes, sir.
Here the only thing it is, this is an inhibition. Inhibition like the first one, correct? Inhibition like this first one. Yes or no? Inhibitor molecule determine the shape of the active site. Yes or no?
Yes, sir.
In that case, it is like the, what is it? First one. But only difference is That inhibitor molecule is whom? Who is functioning as the inhibitor molecule?
Isoleucine.
Isoleucine mean the end product, correct?
Yes, sir.
Are you clear? Are
Yes.
you clear?
Yes, sir.
Okay. In a normal allosteric regulation, an external molecule only will be doing the inhibition, correct?
Yes,
Yes.
sir.
Here, the end product itself is doing the
Inhibition.
inhibition. So that is called by the name feedback inhibition. This is minimizing the work. How? Until I am used up. Now, normally our parents will tell, adikadi saapidade, don't eat very frequently. Okay, the food should digest first, correct?
Yes, sir.
After the food digests, when your stomach is empty, you will feel hungry, then eat. Agree?
Yes, sir.
Similarly, isoleucine has been produced. Until it is used up, another isoleucine is
Produced.
not produced. Understood? Understood my point?
Sir.
Isoleucine has been produced. Isoleucine has been produced for a purpose, no?
Yes, sir.
Until isoleucine is used up, do we need another isoleucine?
No.
No, sir.
No, no. This isoleucine is there, correct?
Yes, sir.
After it is used up, another isoleucine production is reasonable. So until isoleucine is used up, enzyme is inhibited by isoleucine by binding to the allosteric site by changing the shape of the active site. Clear?
Clear, sir.
Clear, sir.
Okay. Right. So that is what we call by the name, what is it? Feedback inhibition. Here, they are not taking isoleucine, no? What is it? 309 for this. They want simple examples for you. ADP, ATP example they take. They say ATP functions as a, what is it? Inhibitor molecule, minimizing the ATP production. Understood? So in this case, who is the end product? In this example, who is the end product? The end product is ATP. Okay. ATP, okay, inhibits, okay, the enzyme until ATP is, what's it?
Used up.
Used up. After it is used up, there is need for ATP, so it should be produced again. When it is present, no need to waste the metabolism and the enzymes to produce ATP in excess. மனுஷன் யோசிக்கிற மாதிரி enzyme யோசிக்குது இல்லைன்றதுதான் உண்மை. Correct? How human think? He earns without spending. Correct? He further earns. Without spending, he further earns. Right? What are you going to do with this money? Don't know, but I want money. That is human nature. None of us are, okay, exceptions for that, including me. Yes or no? But enzymes are not thinking in that pattern. Okay? Enzymes are inhibited. ATP is there. Until ATP is used, no more ATP is needed. Wait until ATP is used. Till then, you are inhibited. Enzyme is inhibited. Clear?
Yes, sir.
Right. In the ரெண்டு point, mark these 2 points. With this one, what is the lesson over?
Metabolism.
Metabolism is over. So, do you know what are the remaining components of the unit number 2? Do you know what are the remaining components of unit number 2? Do you know?
No, sir.
No, I will tell you. I will tell you. Okay. There are these things I have to cover. Okay. You have, this is your resource book. Okay. Remember, as I told you last class, okay, your first term examination, okay, will be demanding to cover your lesson up to Kingdom Protista of unit number 3. How much to cover? How much to cover?
Kingdom Protista.
Kingdom Protista of unit number 3. Okay, we are now in the place. Now we are in the place. Now you have first term examination in March. FWC examination. Understood? FWC examination. ஸ்கூல்ல வைக்கலையா நான் வைப்பேன், ஸ்கூல்ல வச்சாங்கண்டா விட்டுருவேன், ஸ்கூலே செய்யட்டும். ஓகே, right. I have to cover hereafter photosynthesis. Okay, enzymes are over. Photosynthesis and cellular respiration. அதோட சேர்ந்து in the cell division, there's a small part called tumors and gulls. Tumors, cancers and gulls. That portion I have to complete. Understood? So inshallah, I will send you the photosynthesis tube to you. Okay, give me 1 day time. Inshallah, I will send you the photosynthesis tube. Take the printouts and get ready. Okay, and cell biology. Cell biology. In the case of cell biology, I have given you the short questions. Correct? How many? Okay, I think Amira has not got the short questions yet. Humaira got it. Humaira, have you completed Puta? Short questions on. Yes. Amna has completed. She sent me also. Okay. Avasha has completed, I think. She was asking for the scheme. I will send the scheme. I have the scheme. Okay.
Sir.
Right. So after that, on cell biology, you are going to try a structure question series. Structure question series on cell biology. 3 structure questions, 3 structure questions series are there. Part 1, part 2, part 3. 1 by 1 will be sent to you. Okay. First of all, today, inshallah, I will send you the short question answers to you. Okay. Then we will decide when we can hold the first structure paper. Monday will be okay. It won't take much time. Don't take 1 hour. That is not there. அதுக்கெல்லாம் ரொம்ப காலம் இருக்கு. This will be a 15 minutes, so half an hour paper, that's all. Understood? Understood?
Yes, sir.
It won't be taking more time. எதுக்கு டைம் எடுக்கும்னு கேட்டா நீங்க ரெடி ஆகிறதுக்கு தான் டைம் எடுக்கும். Understood? You have to prepare yourself. Okay, in my subject, that is the mode I follow. That is the mode I follow. எனக்கு தொடக்கத்திலிருந்தே புள்ள ப்ரிப்பேர்ட்லயே இருக்கணும். I don't like children getting prepared in the last moment. From the beginning to end, there should be, what is it? Prepared state. That is very, very important. That's what I'm trying. The most successful system is staying in what? Prepared state without preparing in the last moment. So now your results have come. You are getting ready. இவ்வளோ விஷயங்கள் நடந்துட்டதால தான் I have increased your, now you know, we started in August or July, I can't remember. Groupல பார்த்தா தெரியும். Okay, so this moment I was doing only 2 hours class per week, correct? From December onwards only, I have pulled it to, what is it? 4 hours, because time is right to start the 4-hour session. Okay, then only syllabus can be covered on the right time. You can be pushed to the examinations. Remember, okay, எனக்கு ஹுமைராவை அப்படி ரிக்வஸ்ட் பண்ணலுமோ தெரியாது. ஓகே, ஷீ இஸ் கம்மிங் ஃப்ரம் மாவனில் கிட்ட இருக்கிறவங்க. Okay, better you do the exams, what is it, physically in my place. Clear?
Yes, sir.
Okay, we'll meet inshallah. Photosynthesis cute I will be sending you by with tomorrow night. Before tomorrow night you will be getting the photosynthesis tube. Okay.
Okay, sir.
Okay, Allah Bye.
Jazakallah, sir.
Bye.